Hello and welcome everyone to the CyVerse webinar series. I'm Tina Lee, CyVerse's User Engagement Officer. We hope you'll join us every other Friday through December 3rd of this year for our webinars on technology, platform, and science topics. Many of you already use CyVerse and know that we are a National Science Foundation funded cyber infrastructure project. These webinars help fulfill a key part of our mission to train scientists on how to use CyVerse's computational resources to do their research and teaching. First, let's take care of some housekeeping and then we'll start the webinar. Our presentation today is roughly 30 minutes or as long as Amanda takes, with time at the end for your questions. Please be sure to open the Zoom chat window and type your questions there. As always, a video recording of today's webinar will be posted later on our website webinar page for your viewing pleasure at any time. CyVerse offers intensive training such as our foundational open science skills and container technology workshops. For more information on those, please visit our website at www.CyVerse.org for information about these trainings and other cloud-based tools and techniques for collaborative open science. And now I'd like to introduce today's presenter, Amanda Cooksey, who's one of our science analysts on our science team. Amanda does everything from integrating apps to responding to user questions as part of our support team, and does her own science as well. So we appreciate her taking the time to show us some of the key features for how to improve the fairness of your data using CyVerse. So let's begin. Welcome, Amanda.
Thank you. All right, so today we're going to be talking about CyVerse features for fair data. So enjoy this graphic while you can. So this is my apology/warning. This is one of the very few graphics I could get into this talk because fair data, it's very texty. So anyway, this lovely graphic I borrowed from the Wikimedia Commons. And basically it says that FAIR stands for Findable, Accessible, Interoperable, and Reusable. And of course, the goal is to make all of our data fair, which sounds like an incredibly good goal, but doesn't give you any information about what that actually means and how you might go about doing it. So before we get in too deep, I just want to cover some basics. FAIR is actually just a set of guidelines, and there are lots of different ways that you can meet those guidelines, some of which will be perceived as better than others. But it's absolutely worth doing something. Even if you can't do the very gold standard best thing that could possibly be done, the worst thing you could do is nothing at all. A little bit fair is better than not fair at all. So CyVerse provides you with several different features that are really just opportunities for you to make your data more fair. It's still going to rely on you to actually go and enter all of the very important metadata that are related to your particular research, because you're the only person who actually knows that, and we can't do it for you. The easiest way to plan for having fair data is to plan for it at every stage of the data life cycle. So we've all been there. You're going to publish your paper, you're trying to write up the manuscript, you've got to go back and remember everything you ran and how you ran it and what the parameters were and where you got the samples and how old your subjects were. And if you haven't been keeping track of that all along, it can be incredibly tedious, to say the least. And finally, I just want to say, you'll notice this as we go through the rest of the talk, metadata, metadata, metadata. Metadata are really the heart of fairness, even though there are other things that you should do, but metadata go a long way. So F is for findable. As I said before, they laid out FAIR as a concept, but then that doesn't really tell you very much about how you accomplish it. And so in 2016, a group came together and wrote some guidelines in a Nature paper about what the specifics are that we're trying to get out of each of these things. So F is for findable. Metadata are assigned a globally unique identifier, and it should be persistent. Described with rich metadata, clearly and explicitly include the identifier in the metadata of the data that it describes. Metadata are registered or indexed in a searchable resource. So basically, having a globally unique identifier would be the gold standard, most wonderful thing you could do. But having any identifier is better than not having an identifier at all. Rich metadata, everyone should be able to do this because there are always things about your data that people can't tell just by glancing at it. So metadata certainly one of the easier things to accomplish, actually. It just takes some work. And it should be in a searchable resource because that's how we find things. We search for them. So that makes sense. A is for accessible. And this really is mostly about being able to get things. They should be retrievable by their identifier, which you had to have anyway. And it should be by a standard communications protocol, not some strange thing that you can't understand or identify or get. So it needs to be open, free, universally implementable. And it should allow for authentication and authorization where necessary. And for whatever reason, if your data were to vanish from the earth, would your metadata still be accessible? So that's something to consider. Interoperable. Metadata use a formal, accessible, shared, and broadly applicable language for knowledge representation. They use vocabularies that follow FAIR principles, and they include qualified references to other metadata. So a lot of this comes back to the fact that we have so much data at this point, and it's just more and more and getting bigger at an increasing rate, that we rely on computers for pretty much everything at this point. And if you don't use standardized vocabularies and standardized schema and a formal language and standardized formats, then it's not something a computer can interpret very well. And including qualified references to your metadata and other metadata just links projects together. Things that are related should be relatable. People should be able to find one and then link to another and follow the little breadcrumbs along. R is for reusable. Metadata are richly described with a plurality of accurate and relevant attributes. They're released with a clear and accessible data usage license, and they're associated with detailed provenance, which basically just means tell us where it came from. And they have domain relevant community standard, or meet a domain relevant community standard. Some community domains don't have very well-defined standards, and other communities have done a fantastic job of implementing these community standards. If you do next generation sequencing, we have actually a pretty decent setup. I have met people from other domains that have a much more difficult time. But again, this whole thing really comes back to metadata. Tell us what these actually are and where they came from, these pieces of data, because we can't reuse it if we don't actually know what it is. So that's where basic guidelines of FAIR. And before we get into the CyVerse features that will help you accomplish those things, I just want to talk briefly about licensing. So licensing is one of the things that catches most people off guard because it's not really a scientific thing, it's more of a legal thing. And we're not used to speaking that language or dealing in those circles. So this is a link to opendefinition.org, and they have just general information about open data licenses. So if you scroll down, there's a nice little table here that tells you about some of the more commonly used licenses for open data. So there's the Creative Commons CC0 license. And then the next one is the Open Data Commons Public Domain Dedication and License. So you can see they've given you some information about exactly how these work. So Creative Commons is good for content such as like white papers or workflows, something that you would otherwise be able to copyright. And it's also good for data sets. So it's pretty across the board, decent public license. The Open Data Commons is really only pertinent for data sets, but that's fine. That's what most people are publishing through CyVerse anyway. By actually is whether or not it requires attribution. So if you license your data this way, are people required to give you attribution when they use it? And in this case, neither of these requires attribution. And SA is for share alike. And share alike means that in the way that this data was provided to you to use freely and openly, you should provide any derivatives that you make to the general community in the same way. And then in the comments, and this is really important to read because I think a lot of people don't necessarily think about this, you are dedicating your data to the public domain. You are waiving all of your rights. You're not going to patent this. You're not going to copyright it. It's just free for everyone else to make use of. So that's a great thing, and it is what many, 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 probably the majority of people do. So don't feel like you shouldn't do it, but you should be aware of that's what you're doing. Something else to take into consideration is that institutions and funding agencies may have specific licensing requirements. If you work at a public university and you get your money from publicly funded government agencies, generally the more publicly freely available you make things, the better. But there are exceptions to that. If it's something your university wants you to get a patent on, then obviously you can't just dedicate it to the public domain. So you should really pay attention to whether they have licensing requirements on you. Okay, so moving on to some of the CyVerse features. So I've got 2 more texty slides, and then I swear I'll stop texting at you. And this is just a couple features that we have in CyVerse that can help you make your data more fair. And then after this, I'll take you to CyVerse and we can actually look at how you might do that. So Elasticsearch, which we use in the discovery environment, makes it a searchable resource, which of course meets with findability. So if you look out here, at the end of each of these, I provided the FAIR letters that it speaks to. So this speaks to findability. CyVerse, you can apply metadata. This is something that not a lot of people know about or use, but it is extremely useful. So our metadata are in a standard format called AVU, which stands for Attribute Value Unit. And basically, if your attribute was plant height and your value was 12, your unit might be centimeters. Or if your attribute was identifier, then your value might be your DOI number. And you don't have to have a unit, that's optional. We have templates for applying metadata that apply community standard schema and standardized vocabularies. Speaks to interoperability as well. Metadata can include refs to other identifiers. So some of the ways that people make use of this, not that this is an exhaustive list, you can use it in many ways. You might include the biosample ID you got for your sequencing data from NCBI and attach that to the metadata of all your analytical products. You can include the DOI of the publication you wrote about this particular data set in the metadata for the data set, or the DOI of a related data set. Or this is a really good place to put your provenance information as well. It may be the identifier from another resource of the data that you pulled from that resource to start doing your analyses. And that's information that you need to include in some way anyway. And one thing that people tend to overlook when they talk about metadata in the DE, it's not just applied to your files, but also if you're doing your analyses in the DE, then we keep track and associate as metadata with that analysis what app you use, what version of the tool it was, what your parameters were, what your input and output files were, and therefore you know what the formats of those files were. And that can make it much simpler to figure out what you did when it comes time to publish. CyVerse curated data, or DOIs. So this is probably the gold standard thing that you can do through CyVerse to make your data the very most fair. So a DOI is a direct object identifier. It's a globally unique identifier, and it's included in the metadata. That identifier will be included in the metadata for the data set it's attached to. We use the DataCite metadata schema for that, and that's domain agnostic, so it's fairly general, but you can add more domain specific information piece by piece or with another template on top of that. We use the CCO or Creative Commons 0 or the ODC PDL licenses. If you need another license, it's not entirely out of the question, but you will have to contact us. Data and metadata are both accessible via the CyVerse Data Commons using HTTPS, which is about as free and open as you get. Basically, anyone with an internet connection can download these things. For larger data sets, you can also download them using iCommands or CyberDoc through the discovery environment. And metadata are also available at the DataCite Commons. So if CyVerse vanished off the face of the earth, your data would go away because we would be hosting them as a part of the DOI issuance. But your metadata are also hosted through DataCite because they're the people who help us to issue the DOI in the first place. Okay, so the other thing that we have at CyVerse that may be of great use to you if you're doing next generation sequencing, in the transcriptomics genomics type domain, we are lucky to have community agreed upon metadata standards and archives and repositories for depositing your raw data. So the sequence read archive or the whole genome sequence submission pipelines will help you get your data if it's already in CyVerse. Why not submit it from there directly to NCBI? Because it's going to have to go to NCBI. That's where your raw data have to go. So we have submission pipelines that will help you get them into the correct repository. And NCBI, of course, collects huge amounts of metadata and issues globally unique IDs, which you can then include in all of your other metadata. So I think that's all of the features I was going to tell you about. And then I will go to, I need to get out of full screen. Okay. Okay. You're killing me. Hitting escape. All right. So if we go to the data commons, okay, great, there we go. Okay, datacommons.cyverse.org. So you don't feel like obliged to actually follow along with me. I'm just going to show you some things, where things are, how you might do things. It shouldn't be tasking on you. Don't feel like you have to keep up. So this is the data commons page, and the data commons page is where we host all of our publicly available data through CyVerse. So you don't have to log in when you come here. Anybody can come here and get this information. You can browse data. So there's either community release data. And if we click on that, you'll see a list of all sorts of different folders. And each folder really represents a project. And that project is managed by a community group who's requested a data folder. And they have complete control over that. But it's a living data set. They don't have a global identifier that's been issued. They can keep adding data to it as they generate more or updating versions of it, but it's made available to the community. So there is the Dublin Core metadata template, which we asked them to apply to these projects. So there is metadata, but there's not a global identifier. Whereas CyVerse Curated is where we keep all of the information relating to projects for which we've issued a DOI. So all of these are DOI issued data sets. You can see there's quite a few, and you can see Carolyn Lawrence Dill's group does quite a few of them, and they do an incredible job with the metadata. So I'm just going to choose one of their projects to show you. So you can see it's got the project name at the top. The DOI is displayed very prominently, so people shouldn't have any trouble finding that. And if you were to plunk that into Google, you would probably end up here. Creator, description, this is all stuff that they pulled out of the metadata. You can show more metadata. That's still not all of it, but it is more information. You can download the metadata, and that'll be in JSON format. You can also go into each of these folders and see what the files are. And you can, if you select a file, either download it here, or you can go to the discovery environment and download it. If you download it here, it's done via HTTPS, which is not going to be the fastest download, and you have to download it one file at a time, but you can get it. If you go to the discovery environment, you'll get either with iCommands or CyberDuck, you can have a threaded download, and you can download the whole folder in one go. So if you want the whole data set, I would recommend doing that. But for right now, I'm just going to take you back to the front page here. We also have 3 other buttons on this page, and each of these just takes you to the documentation for how to do things. So this will take you to the SRA submission pipeline, same for WGS, or how to request a DOI for your data set. So now if we go to the discovery environment, I can show you, okay, there we go. I can show you how to do metadata. So first of all, just quickly before we move on to another page, if you wanted to search any and all things in the discovery environment, you can do that here with Elasticsearch, and that's what makes it a searchable resource. But if we go to the data tab over here on the left, you can see these are all your data. And if we select from up here, you can see this is my username at the moment. This will be your username when you're using it. And you can see there's things people have shared with you, there's trash, and everyone will have access to community data. So if we click on that, you might recognize some of these things from the community released page on the data commons, and that's because it basically mirrors the same thing. But if you scroll down to Commons repo, curated, these are all of the CyVerse DOI data sets. And so there's Carolyn Lawrence's coffee project, which is one we just looked at. So if I select that and go to more actions, I can choose metadata. And you see it shows the attribute value unit for each of the pieces of metadata that was added to this data set. So that's just what they are. But if you go to more actions again, you can view it in the template. So for a DOI request, which is what this was, we would require them to use the DataCite 4.1 template. So you can see there are other templates. This is for a specific research community. If you wanted one for your specific research community, we could add that, but you'd need to contact us. And Dublin Core is the template we use for community release data folders. And you can see a lot of these relate to the NCBI submission pipelines. But if we look at the DataCite 4.1, we're going to see that same metadata that we just saw on this page, except displayed in the same template where they added it. So you can see they have title, creator. You can add ORCID IDs for people, affiliations, year it was published, a basic description. You can add more people who are contributors to the work. So if you have, nobody works alone these days. You will certainly have collaborators you want to put on here. A long description. This is, a paragraph just describing what the data set is. Okay, then subjects. And I'm going to selfishly devote a few seconds to talking about subjects. When you request a DOI, we recommend you add at least 3 subjects, and basically keywords. Add each subject in a separate field. I repeat, add each subject in a separate field. You can see there's subject 1, subject 2, subject 3. Don't put a comma separated list in one of these fields, because one of us will have to fix it. If it's the only mistake you made, I'm usually nice enough to fix it for you. But if you made other mistakes, that means you'll have to go back and fix it. And it's the most commonly made mistake. So, if you do make it, don't feel bad, you have company. Identifier, this is where we put your DOI number after we issue it, and the identifier type is DOI. This is the license that they use, the ODC license. You would also have the choice of using Creative Commons, but that's not what they chose. Version 1, you can have different versions of data sets. Funding institute, and these are not required fields for compressed data and data format, but they include them because they're good and they like to do a good job with their metadata. So if we go back to my username, I've made an incredibly lame set of data to use as an example. So if we choose Webinar Fair here as a folder, and then we choose metadata. So if you wanted to add just one little attribute, you could do that by clicking Add Metadata. You can see it pops up New Attribute, and then you can click on the pencil to edit it. And you can say height 6 feet. And that's the attribute is height, the value is 6, the unit is feet. If you say done, you can see it's now attached that metadata to your data set. If you want to add metadata in a template, you can again choose the DataCite 4.1 template. And then here you can see required fields have asterisks. And scroll on down. So here's the subject. If you wanted to put a subject in, you can do that. If you want to put a second subject in, you just hit the plus sign next to subject, and it generates a second subject field. So then you can write dog. And if you want a third subject, you hit the plus sign again, and it makes another empty subject field. Cow. Here under rights, this is where you would enter your license information. So you can choose between the Creative Commons 0 or the ODC license. Click out of that. Any license will do for the moment. Okay, and there are all sorts of things that you can add here. Related identifier is one that's kind of important. If you get a DOI for this data set and you use that when you write the publication, once the publication comes out, it will have its own DOI, and you can come back here or tell me to come back here and add the DOI for your paper as a related identifier, just to keep everything well connected. So I am going to, if you hit save, it would apply all of these metadata to the data set, but I'm going to hit cancel because I haven't filled in all the required fields, and it's going to not like that. So discard. But that's how you would add the metadata. And then you would want to hit save very carefully before you went anywhere else. So yeah, that's how you do the metadata. Once you've got the metadata applied, let me go back. If you wanted a DOI, you can again just select the folder and then do request DOI. And it'll basically say, read the instructions and agree to the data commons policy. And then you just click, I need a DOI. And that's going to send me a ticket. Ooh, yeah, it didn't like that, I suspect, because the metadata aren't applied correctly. And that's fine. But what it will do is it will submit me a ticket in our support system that says this person wants a DOI, and I will review your data set and determine that you filled in all the fields that are required and that they're filled in properly. And then I'll issue the DOI. And once we've issued the DOI, it will appear on the Data Commons website over here. So it'll be one of these. And when you search for your DOI, you should get a landing page. That's not the best example of landing pages. There we go. And your DOI will be at the top of your landing page. WGS pipelines, there are instructions on our read the docs for how to do this. It's a multi-step process, and I certainly don't have time to cover it right now, but if you just went to read the docs and search for SRA, likewise, if you come here and just search for DOI, it'll bring you to the right page, but that's learning.cyverse.org. And if you come here, you can get all the little instructions, but it's basically apply metadata, and then you submit through an app and it generates an identifier. And then there's multiple layers because of how the system works at NCBI, but it should be a fairly simple process. And then you'll have identifiers that are community and domain specific with appropriate domain specific relevant metadata. So going back to the slides for just a second. There we go. I included on the last slide some links to useful resources. So this is just a link to that Nature paper where they lay out all the different guidelines. There's a nice little tool that the Australian Data Commons provides for how to assess the fairness of your data. So whether you're at a level of good, better, or best, then you can get some idea of that here. It's actually a really neat tool. This is the page I took you to about licenses, if you want to read more about other specific licenses. Documentation on how to do the SRA and WGS pipelines, as well as the DOI request documentation. And then this is just a link to our user portal. And there you can make requests for a community release folder if that's something that you think you and your community are interested in having. These are not going to be clickable, I don't think, once we publish the slides. So we're hoping to include it in the webinar materials on our webinar page. If not, go to learning.cyverse.org. That's where most of our documentation is, and that will get you a good chunk of this. So I think that's all I have, and I am happy to take questions.
Thanks, Amanda. I will definitely put those links onto the webinar page along with the video recording, so people can access those. There are some questions. Do you have an estimate for the amount of labor involved in the Lawrence Dill level data curation as expressed in FTEs? In other words, how many hours per week does FAIR require? What level of proficiency in data science is required to properly curate FAIR data? That's a lot to unpack in there, but go for it.
So I don't have an estimate in FTE. I will say Darwin Campbell does all their submissions, and he's got it down to a science. So it's like most things, that first one's a doozy. It takes you a little while to get the hang of it, but once you've done one, doing the second one is much easier. It really is a matter of knowing what data you have and knowing about them. And to be fair, you're the only one who's going to have those answers. You're the one who knows your experimental design. You're the one who knows about your research subjects. So I would say it probably take you a couple days to do the first one. But I can't speak to how difficult it is for you to collect all the information. If your collaborators have the information, that makes a difference, right?
I guess my question would be, excuse me, Amanda.
Yeah, you and me both sound like that.
Allergies. Is it easy to edit metadata once you've submitted it, or is it?
So if you're just adding metadata to your folder in the discovery environment, it's absolutely easy to go in and edit it. As you saw, I added an attribute, and then I could easily just go in and click delete, and it would go away. Once you've requested a DOI, that's permanent. That identifier is permanently associated with that particular set of data, not that set of data with a few changes. There are no changes, you're done. So you really want to make sure you've got it as you want it before you get a DOI. There are a couple exceptions to that. You can make changes to the metadata, like going back to add the DOI of a related publication or a new related data set. Or if you need to make changes, because if it's erroneous, we don't want that. If you need to make changes, we can deprecate the old version and make a new version and link them together so that people can get the new and correct information and realize that the old information is no longer valid.
Oh, okay, thank you. Are there any other questions? I see some logistical questions about sliding the shares, sharing the slides, and a link to the webinar page, which I've provided there. So basically what we do after these webinars are done is I go back to the webinar page and I embed the video recording and slides and things like that. I'll put the links into, since as Amanda said, we'll probably PDF the slides. So these links won't be interactive, but we'll provide separate links. And if there are any more questions, feel free to type them in. Okay, can we reach out to Amanda in case of further questions when attempting fairness practices and CyVerse?
So if you've got just general fairness practice questions and CyVerse, send it to support@CyVerse.org. It may end up with me anyway. If you've got specific questions about DOIs, you can send it either there or to DOI@CyVerse.org. You may get me on the end of all of those anyway. So
she's trapped. We won't ever let her stop.
Yeah, it turns out if you want to know something I don't know, you may be in trouble. You always end up with me.
All right. Well, if there are no further questions, join me in thanking Amanda for helping to demystify the myriad versions of what to do to get your data more fair. And please join us in 2 weeks for our next webinar, when CyVerse's Il-Young Choi and Sarah Roberts will do a technology preview and demonstrate how to streamline connecting your data to apps, thanks to their ongoing work on something called the iRODS Container Storage Interface Driver. So that's aimed at general users as well as scientific infrastructure developers. So please join us for that in 2 weeks. And in the meantime, thank you, everyone. Stay safe, and we'll see you back here soon. Bye now.
Bye.
Thank you, Amanda.